And you've got it in sonar. Let me keep coming down here, sorry. And Carl left a note for us that we are on until noon. They're going to try to figure out the static loading of the cable for us.
All right, looks like you've got a possible feature to your right, closer than the main wall. It's small, but
so we are at the bottom. We are going to wait for the ROV team to settle in, figure out what they need to do, and once they've given us the all clear, we'll start science ops.
All right. Yeah, the problem with a face like this is there could be a higher current closer in than further away, or vice versa. So, you know, it doesn't look like there's much right now. So I would just dial it in right here. You can.
So we're back in the land of volcanic rocks.
You can do it visually.
Somewhat
or
sedimented.
You can cheat and put in. You're going the other way. You want to go the other way? I believe. Yeah, there you go. You can put in like auto depth, see what that gives you for percentage, and then yeah, that's cheating, but yeah.
All right, how do you guys feel about a bailout direction of 070?
Nope, that's
that sounds good.
On right now.
Northeast.
Hi, this is Patty Breyer, and down at the bottom center of the view, you can see the interior section of a pillow lava. When we dove in a sort of deeper than this, we were in a field of bonanitic lava in the Shinkai 6500. So it may be that these are exposures of the same sort of thing.
Sorry, Patty, I didn't catch the end of that. What kind of exposures?
That was a pillow lava cross-section. You could see the curved outer portion of the chilled margin of the tube of the pillow. And the composition of things that we found a little deeper, they were bonanitic lava, so high magnesium andesite composition.
Yes.
Hey, we'll be ready to go in just another minute. I gotta get the autos dialed in on the vehicle, and we're ready to start exploring. One note is we were doing a little experimenting earlier. Due to the depth,
so Deb and Dave, I just wanted to let you know that we are again standing room only in this ECC. I had to set up another table.
Brian, I can't hear. Hold on a second.
Accommodate the group today.
I'm sorry. Could you try that again?
Sure. I was just saying, on the way down, we're experimenting with a couple of position moves, and it's going to take 20 to 25 minutes for the ship movement to translate down all 5 kilometers of cable to get us going. So once we get going in a direction, there's not going to be any stop and go back. And if we hold in an area for a long period of time, we're going to be there for an even longer period of time. So we're trying to keep a somewhat very slow, consistent pace is probably going to be our most efficient route today.
What does that say about sampling?
It's going to make it a little challenging.
Okay, that's very helpful. Thanks.
No problem.
I think just to add to that, I think it just says that if we are at a spot where there is a possible sample and the ship is stopped, then we should go ahead and do it.
Got it.
Hey Chris, it's Diva. So great
You're
that
getting, way
they're
out
standing
and
room
way up.
only. Hello, UHECC.
I think you should come back
I think
down.
this is probably one of the most exciting dives, hopefully, on our agenda for this expedition. And so it's we'll see what we find down in the Marianas Trench today.
Go ahead.
Brian, what computer and software do the scientists use for their logging?
That was Roland.
You mean for the event log?
The
Patty Fryer again at the ECC in Hawaii. I'm wondering, are the lasers on? I don't see it.
Patty, we tend to turn the lasers on and off depending on what we're doing. Right, yeah, they're not on all dive, and they're probably off right now because they're just still finishing setting up the ROV. But we can ask for them to be put on when we need to.
Okay. Okay, thank you.
Say again. Okay, we can head on down. How's the current look down here? Okay. I'm going to bring your lower swing arms out a little bit. It's not. The arm's kind of in the way. Don't worry about it. I'm talking to myself. Alright, take a look around. There's something center left of your screen. Top.
Okay, so we have our first fauna of the dive. It looks like there are two holothurians in our view, so sea cucumbers. I can't quite see what kind they are.
So, Patty, you want to tell us a little bit more about what we're seeing right now?
Right, Sean. Still getting set up over there. Okay. Sorry, can you move the mic a little closer?
So while we're waiting here for everything to get set up, Diva, would you like to talk a little bit about the overview for the dive?
Still kind of quiet, but better.
Yeah.
Sure, I'm happy to, unless Jeff Drazen, who's the one who submitted the dive plan, wants to chime in. But if
ready,
Jeff does not, I will more than happily do so.
And Jeff, this is for our for the video that goes along, or sorry, the audio that goes along with video. So even though some of our science people have heard it before, people joining us from shore may not have.
All right. Well,
you can put one
I'll
up
just
there.
give a broad overview then. A lot of the work that's been done in the trench area has either focused on the very deepest part,
I don't know if
the
you can
challenge of
control that
deep,
screen
or it's
from that
focused on the
station.
shallow areas, a lot in the back arc and in other places. And there's a zone between about 5,000 and 8,000 meters that really has been poorly investigated,
quiet,
and that's what we're trying to sample some of today.
I'm going to hold here.
And so we'll try to cover some rocky habitats like where we're at right now, and then over the course of the dive, as the slope changes, we hope to find some a mixture of different habitats, from sediment covered to some more rocky habitats, and look at a diversity of animals.
Fantastic. Thanks very much. I think we found both rock and sediment for you right now.
No problem.
So what's the plan, Nav?
Joe, would you like me to zoom out? Zoom in? What are you thinking? Okay, I'm going to come out. I'll give you full wide.
Can we zoom in on HiPAC? There's a fish to your right, Sean, to your starboard.
So that holothurian was either from the genus Amperima, Peniagone, or Scotoplanes. We're not quite sure yet.
He's above you. I can't see him right now. He's like... I was watching him in Sirius. I don't know where he is. Oh, he's to your port.
So Mackenzie Geringer, a PhD student at University of Hawaii, who works a lot with hadal fauna, she's saying it's definitely not Scotoplanes. There aren't enough appendages on top of it. So it's likely to be Amperima or Peniagone, two genera that get confused quite a lot.
Yeah, I mean, it looks fine. I don't see anything on the Sirius sonar. So. Yeah, do a... Okay, stand by. Yeah, I don't know where he went, Sean. Right, let's come up and push ahead. I just want to see what's ahead of us. Yeah, we could do like a 30-meter
So for the
move.
plan for this dive, we're starting right about 5,000 meters. We're going to be moving up the slope of the trench wall, and we're going to see how far we can get to. We've tried to plan this to have a fairly a steeper slope towards the bottom of the dive track, and to emerge onto a flatter area towards the end of the dive.
You want to zoom on Sirius here?
Sure.
Did zoom on Sirius.
Oh, you want to? I thought you were going to tilt up toward D2.
Nope. There's a fish here. So we're about to put in a ship move at 225 here, 30 meters. Looking at the sonar, I don't really. It looks like a slope here. You copy that, pilot? Right. Come on out on Sirius.
The fish, please.
So you see this fish behind you?
Go ahead.
Serious.
I repeat, range 30 meters, bearing 225, speed 0.2 knots.
There is a fish behind you if you want to chase it.
So we have our first fish of the
bridge move initiative.
It looks like an ophidiid, which is a quite common fish found in, well, common being a relative term, common fish found in trenches worldwide. Pilot or nav, could you let us know what the slope is that we're looking at, the angle of the slope? Great, thank you.
That would be my guess. So we're heading 225. I'm off to the port more. Yeah,
So
if you want.
Patty,
I was just keeping the fish.
Can you tell us a little bit about the geology that we're seeing?
Patty, I'm guessing that most of what we're seeing is lava flows, and it's just slightly sedimented. You can see broken off chunks of probably close to where they were broken off and partly covered with sediment. But those areas between the ridges,
So Nav, I
probably have moved.
think the
down
plan
close
is going to be
some of
to
they don't
try to keep
look
the ship
like,
moving as long as we have a steady
really
slope.
nice
I'm sure Brian talked to you about that.
So I am kind of waiting.
But because of
to
the
see
delay,
if I can figure out whether this is a whole bunch
as
of
long as we don't see any
drools
obstacles, we'll just keep
lava
moving
that
ahead
have
at,
come
say,
down
like
the
30-meter moves.
or
So
whether maybe Bob
as the ship
Stern,
is
who I
finishing
know
its
is watching
move,
these things, has
we'll
got
put in
another
another one.
idea.
So a little more about
Yeah,
that fish
yeah,
we
'cause
saw.
of the delay.
We're guessing
Yeah.
that it is an
That's a good
ophidiid,
point.
and these fish tend to occur sort of in the abyssal plain to upper hadal depths. So from about 4,000 meters, I think, to about 6,500 meters, where they sort of fizzle out. Thank you, Mackenzie, for sending all that information. Yes, Jeff?
This is Jeff. Is the ROV spinning? Can we try to get a better look at that fish that's behind us?
I think they're still.
Yeah, Jeff, I think I saw that fish scooting away. I think it may be too far away now to catch up, if what I saw was correct.
Yeah, I'm trying to look for
Nav,
the fish.
can you just give us a hint when we're ready to go? Yeah, on track.
Video, this is Survey 1. I no longer see the fish in the Sirius view at all.
Okay, well then, when the ROV is ready, let's slowly start making our way up slope.
Do you guys catch that? So when we have a moment, we're going to try to work our way up slope. Okay, great.
This is Patty. I'm just wondering if we could pick up that rock that's in the center of the center.
That's the ship, Patty, we can't right.
The lasers are on right now. Is that too big?
I think there's a couple problems. I think one, it actually is probably too big, but they're also trying to get the ship underway, so we're going to start moving right now. So I don't think we have time to stop and take a sample. For
okay.
Oh. Okay. Okay, so, Patty, we might have 10 minutes to take a rock sample. But remember that we only get 2 samples for the whole dive.
Right,
So
but that was a pretty big one.
That big one that is now out of view, I think that might have been too big.
Yeah.
But is there anything else in view now that looks like something you might want?
The one that's just below to the left of the 2
Littler rocks?
Yep.
Sort of bottom center right now?
Bottom center, yes, thank you.
Okay. So, pilot, could we try to grab that bottom center rock? I believe so.
That's pretty big.
Or the one above it, I think.
The one above it's a good size.
It'd be better to go for the one that's real close to the lasers now. If it's possible.
I can't see the lasers.
Center screen.
Okay, they're in the...
Thank you.
Yeah.
I know I can't see them on that screen at all.
And...
Dead center.
Yeah, it's pretty much in the center of the field of view. It's the arc on the upper left is very light
Zooming in.
on the lower right. And if the manipulator goes for it, a little bit to the left, now, left.
I think Patty's looking at the one to the left of that. That one is too big. What about the 2 that are above that rock?
Zooming up.
Oh, the one to
What
the
about
left is too
those
big.
above it? The one that you were looking at was too big, Patty. We might be able to grab one of those 2 that are in a line right above it. Yeah, that set of 3, exactly.
Okay.
Would that be okay?
Yeah, I would go for the one the lasers are on right now.
Yes.
That's really big.
That makes the rock about like this.
I think you keep picking the big rocks.
The bigger one
Yeah,
would be...
Could we try to get the one below that, maybe?
Yeah, you can right, one of those 2 would be okay.
Yeah, the one the lasers are on now. Would that be all right?
That looks good.
Okay. Let's double check that.
Yeah, I think we should go for that one if we can.
Copy.
Okay, when the manipulator goes to
Any of
grab
those 3
those,
would be a good size.
if those rocks are off,
Yeah, it's a little big. It's on the bigger
then they're
size.
sediment and we don't
Your hydraulics are up.
So, I'm sorry, can you say that again? You're coming through very quietly on my headset, so can you repeat that?
Is this better?
Not really.
Really? Okay.
Okay.
What I was going to say was that as the manipulator grabs the rock, if the rock is the sedimentary rock, it may crumble.
Okay.
And if it does, then we don't want that rock.
Okay. Perfect. All right, I think we've got it now.
So give it a good squeeze. See if
I'm
it
sorry,
crumbles.
pilot, did you ask me something? Okay.
No, thank you. Sounds good.
Patty and Shoreside, this is Roland from video. Just letting you guys know I am adjusting audios from the phone line as well as the scientists here, so just continue to talk to your normal level and I'll adjust here on the ship.
You want it on the big screen?
Okay, thank you. I'll stop shouting.
Diva and Deb, we have a question about the lasers, and I'm just trying to remember exactly what the spacing is. Can you confirm the spacing?
10
of the lasers,
centimeters.
please?
Yeah, it's a 10 centimeter laser, the 2 spots.
Tilting down. Okay. Port box. Coming in board. Tilting your mini Zeus up.
All right, trying to pick up our first rock. It's an exciting moment.
Yeah, I'm moving now. Nav.
Okay, so port forward.
Pretty sure I'm moving now. Yeah, they want you to give it a good squeeze just to see if it crumbles.
That looks pretty hard to me.
Oh, no.
Take that back.
Yep. All you got to do is squeeze it and nothing happens. Let's see.
I don't think we want that one.
No, I don't think so either, Patty. If they're all the same, we may have to look for something else.
I think this is probably manganese-covered sedimentary material, so forget it. Let's go for a different one.
Looks like it all fell off that ledge.
You know, Patty, a fish wouldn't have done that.
No. One is Zeus, one is mini Zeus.
Yeah, Chris, the fish wouldn't put up with this.
They drop it.
Yeah,
They want to drop it.
we don't want
That one
that
looks
rock.
better.
Okay.
Truly, just,
Yeah, we don't want that one.
yeah. Sorry.
All right, Patty, was there anything else in this area that looked reasonable? Other than the giant one in the middle?
I think not
Okay.
just the giant one, no. I think all of the light material that we're seeing underneath the black coating, I think the black coating's manganese, and we're looking at exposed sedimentary material.
Okay.
Pilots, you can get going when you need to.
Yeah, set.
All right, so Patty, we're going to have to wait on a geosample because we're going to have to start moving. Oh, yeah. I thought we were moving.
No problem. I think we will be.
So, Jeff and Mackenzie, do you think that disturbance will attract in scavenging
There's the two. No, there's one.
Ramping down. Yeah, mini Zeus back. Yep. Well, at least for tilt. I haven't tried to rotate, which I'll try right now.
Sorry, Bruce. We're playing around with the speakers in the room right at the moment. But no, Mackenzie and I don't think they'll be large enough to attract anything. We need either the scent of bait or something bigger.
Yeah.
Oh, yeah, that's disappointing. Maybe we'll see some stuff later.
I'm not seeing a whole lot on sonar, but push ahead. And I mean, it's hard to tell with.
Something
Guys, there's a fish
there.
just on our left.
Fish, stop for the fish.
Yeah, right.
See you there, pilot.
Oh, they're doing it.
Awesome job.
Zooming in. Holding. Zooming in.
So I'm getting IDs that this fish is an ophidiid, as we saw before, potentially. And it's likely from the genus Basazetus, of which very few have been collected. Maybe. I know. Can never be confident if you don't have a sample.
I've got a little more.
Another candidate is that it could be a genus Porogatus. But this one looks different to those that have been seen in those Basazetus that have been seen off Hawaii and Johnson. So it's a lot of the scientists on shore are rethinking their previous identification. The terrain is just so dramatic.
Oops, it is pretty fantastic.
That was great, thanks.
I think we put another ship move in. 30 meters.
All right, so I think we're going to continue upslope.
So Jeff and Mackenzie, was that a new fish record for the Marianas?
Does the bridge go ahead?
Would you like to let us know what fish have been recorded from the Marianas?
5 degrees at 0.2 knots, please.
That's 30 meters, bearing 225 degrees, 0.2 knots speed.
Good copy.
Coming up.
How do you feel about deploying your upper swing arms?
Jeff and Mackenzie, I'm not sure if you heard me, but...
This is Bruce.
Bruce, yep.
Yeah, I would say that request for what fish have been recorded from the Marianas is too big a request right now. We'd have to start flipping through a lot of references. None of us, to my knowledge,
Nav bridge, move initiated.
Jeff has done this, have actually compiled a list of just the deep water fish from the Marianas alone. I've got some lists of fishes reported from all of the U.S. islands, but I'd have to start going through all of those to pick out the Marianas records, and that would take a few hours to do.
So, Bruce, there have been many fish recorded from the Marianas Trench at these depths? That's what I was asking about.
Okay, that's a different question than what you asked before. I don't know, maybe Jeff can answer. I don't think there have been many, but I don't think there's been much sampling. Jeff would probably has the most extensive sampling record here, so yeah, he could answer that. I think he and Mackenzie.
Yeah, that's what I was getting at. I was under the impression that not many had been observed in the Marianas Trench at these depths.
Roger. Survey 1 and Survey 2.
Jeff, Mackenzie, care to chime in?
I don't know what it was at.
Yeah, so Jeff mentioned earlier that there's kind of this undersampled region, that we have several dives from deeper in the trench.
I mean,
and then
that's what it's started at.
But that area between 3,000 and 7,000 is really poorly sampled. So we do have a few lander deployments that have seen ophidioids like that species. But species of the genus Basazetus or various varieties and others. And then we see deeper in the trench a lot of lepterids. So this is a different family that we probably won't run into today. We're a little bit shallow for them. but we might also see mercurids, such as Corzanoides kenna or Yukine, would be pretty common in this area.
Thank you very much, Mackenzie. That was great.
Little jelly.
Zooming in.
Or mucus.
Holding. Yeah, sure does.
That looks like a discarded larvation house. These are gelatinous plankton, and they form a filter house around them. And deep sea ones can get quite large, 10 or 20 centimeters across. And they basically just filter fine particles out of the water. And when they're done, they scoot out of the house and they build another one, and they end up sinking to the deep sea floor as part of the deep sea's food source.
Yeah,
great, thank you,
Jeff.
Jeff.
They were also on like a ridge thing too. It's a little strange. The white thing off to your port. I'll give you a little more leash.
There's something right center to the left. If we could take a look.
Yeah, high voltage
Yeah,
is
Bruce,
at
we spotted it and we're heading there now.
37, which could be anywhere between like 30 and 45. No, that's where it's been. Just under 40, which, I mean, especially on that one, you're looking at like a 10% plus or minus, really.
Pilot, can you give us a head up when we think we can stop for a sample? Just in general, not for right now.
Holding.
Or research.
So this is another Ophidiid. This one's of the genus Lucicorus. Bruce, I think he might be able to talk about this one a little bit more too. We ran into this last year in the neighborhood of Johnston Atoll at a similar depth. I don't know if Bruce wants to talk about the reduction in the eyes on these guys.
Yeah, there are several things to say about this. First off, Mackenzie mentioned the eyes. The eyes are actually very large on this, but they don't have a lens. They're not image forming. Keep in mind that there's no sunlight down at these depths, so this fish could probably detect light, and maybe the eyes are even adapted to detect dim light, but they can't see images like we see with the sunlight. What they may be picking up would be occasional flashes of bioluminescence, which would be light produced by other animals that might signal either potential prey or maybe a predator approaching. This is a good example of the problem of finding out what's at the Mariana Trench from previous distribution, because this is the first record of this genus from the Mariana Trench, as far as I know, unless Jeff has some. We saw these in Hawaii, and it was the first record of the genus from the entire Central Pacific. Otherwise, it's known from some collections in the Atlantic, in the Eastern Pacific, and down in the Solomon Sea, off of New Caledonia, between New Caledonia and Australia. There hasn't been much sampling for these deep water fish in areas except for the New Caledonia area, the Philippines, and Japan around Hawaii. And to my knowledge, aside from the drop camera work that Jeff has done and other people in the trench and on the slopes of the trench, there really hasn't been the trawling work or other types of sampling to let us know what these fish are in the Mariana. So you're getting the first look, first record of this entire genus of fish from the Marianas, and it illustrates how little we know about the fish fauna of this area.
And I think already we can say that we've traditionally underestimated the importance of these Ophidiids, these cusk eels. Those are one of the last ones mentioned in several of the collections because they're hard to catch, but we've already seen 3 today, so very important, underestimated.
Yeah, I think I'll add to what Mackenzie said and just say that a lot of the work on sort of deep abyssal depths has been conducted in more temperate areas of the oceans, off the North Pacific, off California, and in the North Atlantic, off the UK, and the eastern seaboard. And those faunas are dominated by rat tails. But when you get into these more central ocean basins, oligotrophic areas, then the ophidiids really seem to become the dominant group.
How's that light looking? Video?
Yeah, it's fine for now. Thanks, Josh.
Because I could play around with it a bit more if you have a hot spot.
I think it's fine for now.
White thing.
So, pilot, if possible, as we're sort of skirting this terrain, if we could zoom a little more, we don't have to move the vehicle, but if we could just zoom a little more so we can get a closer look to see if there are any smaller invertebrates on the actual rock face. Thank you.
Okay, zooming in.
Can we take a look at that white algae?
Sorry.
So right now we're seeing generally, I would say at least a bit more sediment than we've seen in our two previous dives. Of course, we're also thousands and thousands of meters down deeper.
No, I think we can just sort of move along, but keep sort of zoomed in, yeah, so we can see a little more. Is that possible?
I'm going to disagree with that a little bit.
Okay.
Because we're going to want some wide-angle shots so we can see the geology as well. So it kind of is going to have to go in and out.
Yeah. Okay, that's fine.
There's another white object to the left of center on the top third of the field of view. Can we take a look at that?
And as we're panning away from it, it looks like there are some fractured pillows that are just leaving the screen right now. Bob Stern is chiming in from email and saying that perhaps we're looking at some dikes.
You're right. These could be crabs.
What did you say about Bob Stern's comments?
Bob said that we might be looking at some diking.
Ready for zoom when you are.
Diking.
Sure.
Interesting.
Zooming in.
I'm going to give you some more light.
Oop, holding here.
So that looks like a munidopsid squat lobster,
Yes.
perhaps?
Yeah.
Yeah, I'd definitely say Munidopsis genus. And these are found in all of our world's oceans, pretty much.
Full zoom. That's better, Josh. Were you just rotating the lower swing arm up? Yeah. That's a cool shot.
Maybe not the Southern Ocean, but most of the others.
Yeah, we're moving now.
This is great imagery. Thank you, pilots. So, Jeff, Mackenzie, seen any Munidopsids around this depth in the Marianas before on your previous expeditions?
Let's hold it here for a second or two.
Thank you.
Oh, okay. So Jeff is saying that he thinks this may be a molt of Immunidopsid, which basically means it's when it sheds its exoskeleton to grow. This may have been a... But do you see movement?
No.
Okay.
When you're done with this, there's another white object to the left of this. Maybe
Oh, yeah. Oh, wait. Go, zoom back in. Pilot, can you zoom back in? Just to the left, there's something hanging off. Do you see the thing hanging off of the ledge? I think that's an animal. Just like an...
just about,
yeah, just in the center.
Right where you were.
Are we ready, Carl? Okay, zooming in.
What is that?
I have no idea. Yeah, look at it. Wow.
What is it?
So I think that must be a type of isopod, which is another type of crustacean.
Zoom in a bit more when you're ready.
Isopods are also the sort of pill bugs that you get on land that live in wood, but here's an obviously extreme deep sea version.
Where is its head? Are those its legs?
So, yes, so it's facing us right now, and those two very long things may be its antennae.
It looks like it's something out of Star Wars.
Yes, so a lot of the inspiration for movies like Alien and other things are from deep sea crustaceans. If you live down there, you'd look weird too.
Yeah, James Cameron, who's one of the 3 people that have ever gone to the bottom of the Marianas Trench, and also, made movies like Avatar and The Abyss. Used deep sea animals as the inspiration for the things that you saw in The Abyss and Avatar.
Exactly.
Tilting down.
Can we get the lasers on as you pan out, just to try and get? Oh.
I'll zoom out a bit.
Tilting up.
Holding.
Oh yeah, okay. So that's pretty sizable. I mean, that's a pretty big isopod.
Are we here for a few more minutes, or are we going to keep moving?
Yep. Whenever you're ready, pilots, we are good to go.
Okay, zooming out.
Thank you very much for both those beautiful shots.
Have your camera back.
Bring up what?
On B.
Oh, sorry.
What do you think?
So we have some fissuring, got some fractures.
This is Patty. I'm just wondering if those are cooling cracks.
What kind of cracks?
Cooling cracks.
Yeah, so when it's contracting, basically? I can't hear her.
Center.
There's no
Swimming, dude.
parallel to one another. Oh, worm.
Zooming out.
I think he was heading down.
Was that a worm or a holothurian? It looked like it had tentacles at one end. I don't know what it was.
I don't know.
Definition from a geologist than anything.
And that was a question from the fish geologist, so I don't know. That was a genuine question.
Yeah.
Zooming in. Holding.
Go ahead.
Yep.
I repeat, range 30 meters, bearing 240, speed 0.2.
So, Patty, in addition to the sediment cover, are we also seeing a manganese crust on all of these rocks?
Yeah, that's my guess. Certainly everything we picked up here with ROVs or subs has had some manganese on it, unless it's right in an active spring on one of these volcanoes. One of the interesting things I thought I saw just a moment ago, kind of lower to the left, looked like a cross-bedding contact, whether it was or not.
Your lower swing arms are only partially deployed. Something in the water column to your right, coming at you.
EXRV. So when we stopped just a second ago, zooming in on that white, perhaps bit of trash or something, we did see some. Oh.
Holding.
So that's a holothurian swimming,
very small one.
And you can just see his gut in the middle of his body because he's transparent.
Can we kill the lasers? Thanks. Zooming in a bit more. Holding.
So again, that looks like either Peniagone or an Amperima. And it doesn't seem to be actively swimming, just kind of gliding.
Okay. Zooming out. Okay. Zooming in. Holding.
So I have no idea what we're looking at geology-wise, but there is a holothurian slightly to the left, exactly.
Holding.
You're way up.
Yeah.
So that's...
Patty, again, with regard to the surface texture, looks to me like the angles in these are reminiscent of conjugate faulting, but such a small space, I really can't imagine.
There we go. That's much better.
So that holothurian may be of the genus Benthodites. You can see it had tiny, tiny little projections coming off of its body closer to the underside of the body, but not like podia or feet or feeding appendages. Yeah, so it's sort of dippled with... No, maybe not Benthodites. I have never seen a holothurian like that before. It is definitely a holothurian. You can see the gut just a little bit through the body.
Yeah, we're all kind of stumped with this one.
Right? Okay, pilots, whenever you're ready to move on. Oh, and just to the right of the holothurian, you can see what looks like a little sort of fan-shaped triangular upside-down rock, which is not actually a rock. It's called a xenophyophore, and they are single-celled animals. Oh, and there's a beautiful one just to the left as well. So there are two in shot right now. They're called xenophyophores, and they are a type of foram. They're single-celled, and they build these sort of houses that they live in. Now, these ones are definitely of the plate form. And we see a lot of them in most of our world's oceans, but a lot of them in the abyssal and deeper regions, I think.
That's tough.
And they also go to full ocean depth. They have been recorded at Challenger Deep. And they're just remarkable little, I don't even know if you can call them an animal, single-celled. They're pretty interesting. Great, thank you, pilots.
Yeah, no, I've been watching that. It stepped down a couple minutes ago.
So pilots, the ship has come to a stop, but Sirius will not be moving forward for about 7 minutes. So I can call another ship move whenever you're ready. Sirius actually hasn't started, well, it probably hasn't started moving with this one yet. We have 5 minutes till it starts moving. Another 8 minutes of move after that. But we don't have anything stacked up behind that. Okay.
Yeah, I'm good with another 30.
Copy that.
That we're 60 out.
Okay.
So the other thing we could do is, just thinking out loud, if we do like a 0.1 or a 0.2, is no, that's a lot of tracking. Get confusing pretty quick.
What's that?
We can just do another 30. I'll discuss it later with you.
Okay.
Might be able to work out a better system.
Ooh, this guy.
Crinoid.
Oh, nice.
Standing by.
Go ahead.
Get closer.
Okay, zooming in.
Stand by.
Oh, stand by. I'm sorry. I'm going to turn down nav here.
So
ship move, range
this
30
is
meters, bearing
Brazendid,
240,
which is a type
speed
of
0.2.
sea star or asteroid.
Nav, bridge, move initiated.
Okay.
I don't know if Chris Ma is watching, but I'm going to guess that it's
Frame it first.
either Friella or Friastera. I just need to, how many arms does it have? 6 arms?
Yeah, Diva, I think you're right. I think it's Friella, but hopefully Chris will chime in. And
No, so I think it's going to be Friastera.
our guess is necessary.
Yeah, I'm thinking 6-armed is Friastera, and I think Friella usually has more arms.
Great. Hold it here for a few
But
seconds.
yeah,
and we'll
it'd be
zoom in more.
great, Chris, if you're listening, if you could chime in at this point.
Zooming in.
Do you want me to tilt down to the center?
Sure. Zooming in.
Okay.
Yeah, we've got a Friastera species from the Northwest Hawaiian Islands last year that has 6 arms and looks very close to this.
Yeah, there's also, Chris has also one recorded from the CCZ, which again looks very, very similar to this. Great, thank you, pilots. Whenever you're ready to move on, we're happy to. This imagery is stunning.
Has there been any work to investigate the ground fault on D2?
The initial.
Okay, zooming out.
I don't think so. That'd be
Do we think it's just that the initial GFM is low?
Yeah.
Okay.
Full wide.
At least the couple times I've been involved, it starts at 25 and goes down, 1 or 2 for each.
Yeah.
Like the CTD drops 2, the craft drops 2.
So we're heading at 240.
Is
Just a little more to your port.
Yeah. Yeah, I had gone over starboard for that guy.
The master's at 2 o'clock. They're both at about 2 o'clock. 3 o'clock.